So my role is really focused on microbiology. As you can see from this new beautiful lab, we now do all sorts of experiments related to monitoring bacteria and pathogens, viruses as well. We are definitely setting up more experiments in the area of water and wastewater treatment. And we're trying to sort of expand into other areas to support the PFAS research in terms of bioremediation and so on. I like to pride myself on trying to communicate science in a way that helps our clients make the best decisions possible. The most interesting and probably the most exciting area, and that's the reuse and desal area. As you can imagine, when we talk about potable reuse, the most important part is making sure that water is safe. So we have lots of really cool projects, starting with the Los Angeles Department of Water and Power, Metropolitan Water District of Southern California. We're looking at the product of these advanced treatment plants to make sure they're safe and virus-free. I think a lot of people are still asking the question of, what are the regulations? Are we making sure that water is safe? And I think California is leading the way on that right now. When it comes to scarcity, I think we're at a point where nobody can deny the need in terms of water reuse. Where I think we as CDM Smith and as the water sector are making a huge difference is, this isn't new to us, right? We've been reusing non-potable water for a very long time, for agriculture, for industry. Where I think the major challenge comes in now is that science communication piece, where we can explain to the public that we are actually doing all the testing we can to confirm their water is safe from organics. We're looking at viruses, but we're also looking at cryptosporidium removal, which are protozoan oocysts, right? They're very resistant and you cannot chlorinate them. So we actually do look at the impact of removal, ensuring that everything is removed in that train. And I think ultimately there's also a lot of questions about the distribution of reused water. So you want to make sure you don't have regrowth, you don't have recontamination of that water once it leaves the plant. And that's another area where we as sort of an industry are trying to tackle the monitoring tools we already have available to make them more timely and cheaper and more efficient and field friendly. I actually oversee Water Research Foundation Project 5209, Give Membranes the Credit That They Deserve. And this study is specifically looking at low pressure membranes, so that's microfiltration and ultrafiltration, and how we can help our clients actually get credit for them in reuse scenarios. So right now, if you're looking at most of the regulation around the country, membranes don't actually get credit. And the reason for that is we don't have a good way to do daily testing to confirm that the viruses are actually meeting their log removal targets. So what we did here at CDM Smith is we introduced a portable, field-friendly PCR, and with a variety of adjustments, we're able to reduce the time it takes from potentially 12 to 14 hours to run for 5 targets, all the way down to 5 hours. So in practice, that would mean if you have a plant somewhere in California, an operator could potentially collect that sample and run the qPCR by the end of the day without having to go home unsure what the quality of that water is that is reaching the customers.